Okay, that's it for protein basics. I know this is basics, but it's very complex. Transcription, translation, even the operon stuff, it takes a lot and it's complex, it's long processes. So revisit it a few times. Your teacher will teach it to you as well. Look at different resources. And after a while, it'll just sit in and it'll click. Okay, now we're looking at DNA manipulation. So those are our kind of basics of protein, how we create protein in the cell, how DNA kind of works. Now we're thinking about manipulating this process. So we have DNA polymerase, DNA ligase, and endonuclease. So these are our important enzymes. Don't really worry about helicase too much. So DNA polymerase, you don't look at the intricate details of actually synthesizing DNA, so don't worry about that at all. You just have to know what DNA polymerase does. Think of the name, DNA polymerase. What does RNA polymerase do? It makes an RNA strand. DNA polymerase, it makes new DNA, basically. DNA ligase, think of this as like ligating something. It's basically glue. So it actually joins the backbone. So if you remember our little diagram, we had our phosphate and our sugar. Ligase attaches those. Those are very strong bonds. Remember how we talked about our little hydrogen bonds? So between our little nitrogenous bases, these are hydrogen bonds, pretty weak. That's why we can separate them, unzip them, all that sort of stuff. Between our actual, like our sugar phosphate backbone, so what's highlighted here in green, these are very strong bonds. And that's why we need a DNA ligase. We need an actual enzyme to help us because they're very strong. Don't worry about Okazaki fragments or anything like that. It's beyond the level. Endonuclease, this is really important when we talk about DNA manipulation. Just our little scissors. So if DNA ligase is our glue or like our sticky tape, endonuclease are our scissors. So this is what cuts DNA. Okay, so going into this a little bit more, basically they can cut them with different ends. So you can cut them straight down the middle and this will produce blunt ends, or we can cut them a little bit jagged, I guess, and then we get sticky ends. So in terms of why you would have different ones, it just depends on the endonuclease. So here are our lists of endonucleases. You may also see them as restriction enzymes. They're just the exact same thing. And they have their own recognition sequences. So this is like they're literally programmed. If they see DNA like this, then that's where they'll cut it. So you can see if it's down the middle. So like if you've got 2 on the left and 2 on the right, then it's going to be this blunt end. If you've got, it's like cutting kind of 1 on the left, whatever that is, like 5 on the right, it's going to produce this sticky end. So in general, when we think of DNA manipulation, our sticky ends are a little bit better because we've basically got little like tags, I want to say. So think about our complementary base pairing. AA and TT, they're only going to pair with TT and AA, right? So if you were to chop the DNA up, you've got 2 ends like this. Those will reform very specifically. AA and TT, will not reform with another sticky end that is CGAT, right? Because it's not complementary. It'll only pair with TT and AA. So therefore, when you're manipulating DNA, sticky ends are often a little bit more favorable because you know that they'll be like sticking together properly. Whereas blunt ends, because there's no like overhang, they can attach to anything. So like say we've got this, so GG and CC, these blunt ends, they can stick with anything. Doesn't matter what's on the left. It could be, CC and GG, or it could be A, T, CC, it could be C, U, whatever, not U. But yeah, so that's why blunt ends can be like, I guess a little bit worse, but that's just something to be aware of. Okay, so CRISPR-Cas9, this is kind of a complex system that we look at. So it's a naturally occurring thing that happens in bacteria, and basically we can use this system to manipulate DNA. So CRISPR-Cas9, this will make a lot more sense, or I think it helps a lot when you learn it after you've learned about immunity, like human immunity, because basically this is like a bacteria version. So in general, how CRISPR works is you have a little bacteria. a virus attacks it. Obviously, like, oh, that sucks, right? So what the bacteria will do is, okay, they're going to form their own sort of like immune system. So they will basically take the virus's RNA or DNA. So you can see, to attack a bacteria, these are our little bacteriophages, so they're viruses. They insert their genetic material into the bacteria and then obviously creates more viruses, and that's not good. So the bacteria takes a little bit of this genetic material and it stores it within its own genome. So it's like, it just takes like a little bit of, it's almost like taking a photo, I guess you could say. So like it takes a little photo of this virus and it stores it in its pocket so that the next time this virus comes along, the bacteria will be able to pull it out of its pocket, compare it, and then kind of mount a response really quickly. That's how it uses CRISPR-Cas9. So we take our little photo, our little snapshot of some of this DNA, and we basically stick it into the bacterial genome. So when this virus comes again and attacks the cell, the little, genetic material, which is stored in our CRISPR sequence. So our CRISPR sequence is basically like our little, if we're thinking of these little viral things as photos, CRISPR is like our photo album. It's where we're storing these photos of our different viruses. So we have our CRISPR sequence. Basically, our little virus comes along again. What the cell does is, because, we've got our photo album, we basically take out this little photo, we transcribe that genetic material. Yeah, we transcribe this genetic material, and then we use Cas9. So Cas9 is an endonuclease. So remember what we've just talked about, our scissors. Its job is just simply to cut. So we have our little genetic material. Can you kind of see the way this is going? We've got our genetic material that matches the viruses. We've got our Cas9, our little scissors. What this system does is it goes to this new virus that's inserting its DNA. Cas9, our endonuclease, recognizes it, cuts it. The virus cannot replicate. And that's how it's like a bit of an immune system, if that sort of makes sense. So yeah, Cas9 basically detects that because the DNA is complementary, and then it just cuts that. And so you can see the virus can't attack the cell anymore. So that's how it works naturally in bacteria. What we've done is kind of looked at this and used it for DNA manipulation, because basically if you can program Cas9, instead of looking at, a gene sequence that matches viral DNA or RNA, it can match perhaps like a gene that causes cancer or something like that, and then Cas9 can cut it, and then now that gene is inactivated. So you can see how that's a little bit of DNA manipulation, essentially. Hopefully this might make more sense. So this is our enzyme. This is our guide RNA. So you can see when the Cas9 basically meets RNA that's complementary, it cuts it. And so in terms of talking about DNA manipulation, you could insert this gene of interest. So like, I don't know, for example, like if you wanted, let's talk about it in the context of plants or something, like maybe this gene will make the plant grow really, really big, or the flowers will be really red or something like that. So you can cut it, that's Cas9's job, and then using our other little techniques, you can insert a gene into that little gap. Okay, so moving on to other stuff, we've got this idea of DNA profiling. There are 2 steps of it, and you have to know these processes as well. So PCR and gel electrophoresis. So I'm sure you'll be pretty familiar with what PCR is. In terms of its steps, it's not too complex, but you do have to have a couple of things to start off with. So the point of PCR, polymerase chain reaction, we are amplifying DNA. We're starting off with a little bit. So like, talking about like our COVID tests, like we've gotten a little bit of like this viral DNA or whatever's, in your little nose swab. And we're going to amplify it. So we have heaps of it. And then we're going to look and see if it's, COVID DNA. Or like if you're at a crime scene and there's a drop of blood, you've got a tiny bit of DNA, use PCR to amplify it. So now you have heaps of it. And then you can analyze it and like find out who the criminal is, whatever. Okay, so to do this, you need a couple of things. So DNA primers, so basically just little short bits of DNA. DNA nucleotide. So obviously if you're going to amplify DNA, if you're going to like synthesize DNA, I guess, you need the nucleotides. And Taq polymerase. So this is basically a specific DNA polymerase that allows you to do it in really hot conditions, because we'll have a look at the steps of PCR. Basically, you can see that it goes up to 95, 72. It's pretty hot. So you need to use a polymerase that can withstand that heat. So this specific one is what works. So, okay. Basically, we've got our initial, I don't know if we have we have a diagram here. This will make a lot more sense after I talk through the steps and then you look at this diagram. But you can see you start off with one, you can end up with like a billion strands. So firstly, the PCR mix, so what we've talked about, you heat this to 95 degrees and that denatures, it basically splits the double-stranded DNA into two. Remember, it's our hydrogen bonds. They're not that strong. So if you just heat it nice and high, that is what splits it up. So you can see here, double-stranded, you heat it really nice and high, you split it into two. Then we want our DNA primers to kind of come along. So you cool it to 55. If you were to keep the temperature really high, obviously you can't have complementary base pairing. The hydrogen bonds won't form. So you cool it a little bit back down, so 55 degrees. And this is where our primers, so think about what does a primer do? It basically starts something off. It's like the foundation, right? So our primers come and they attach to each of these little strands. And that's what will kind of get the ball rolling. You then heat it back up to 72 degrees. And this is when Taq polymerase can just work really nice and fast. So you turn it up a little bit and your Taq polymerase, remember, it's just a DNA polymerase. It will synthesize those DNA strands. So it will bind to the primers. It'll start off there and then it will go on and just synthesize as normal. It'll bring the complementary bases and form a new DNA strand. So then you can see what we've got here. We've got now double the DNA. We started off with one DNA strand. Now we've got two. So then we repeat that process again. So we denature them. Now we've got 4 strands. We attach the primers. We've got Taq polymerase. So now we've got 4 pieces of DNA. So we had 1 piece of DNA, then we had 2. Now we have 4. Next we'll have 8, so on, so on, so on. So after a couple of rounds, you can see that just increases exponentially and then we get heaps of DNA. So understand that these are the 3 main steps. You have to mention this idea of repeating it. If you write in your short answer, 95, 55, 72, that's it. It implies that you only did 1 cycle. So if you had 1 piece of DNA to start off with, you had 2 at the end. Like, that's not what PCR is meant for. You want to amplify it. You want heaps. So you need to remember to write about repeating this cycle. Okay, the last thing is gel electrophoresis. So basically this will determine the size of DNA and it creates your DNA profile. So there's a lot of applications. Again, I'll just talk through the process generally, like you'll learn about applications and stuff like that. But it's basically so you can paint a picture of what the DNA kind of looks like. Because technically what you're getting is this just shows what size DNA strands are. But if you think about using like human genes for this, like if you use my genes compared to, my mum's or like my siblings, stuff like that, you'll all have different ones. And so that's how you can use this to kind of identify certain genes and things like that. So the basic principle is you have this little, it's like jelly. That's the kind of consistency of it. So you put these little, like you put the DNA in the wells at the start. And because DNA is slightly negative, remember this, DNA is slightly negative. So I think it's like the phosphate group, something like that. It will move towards the positive side. So electrophoresis, that's where the electro part comes in. So they start off at the negative side, and because, negative attracts positive, they're going to migrate towards the positive electrode, and that's where we're going to get this separation. So think of this jelly basically like a sponge. There's little porous holes. So if you are a tiny short piece of DNA, you find it easy to just weave in through these holes and to wiggle through, and you get to the end really quickly. If you are a longer piece of DNA, you are going to have to, you're going to move through these holes a little bit slower because there's more of you to get through. So therefore you're going to not travel as far. And that's what the separation is. So you have your longer bands towards the start, basically, and your shorter bands towards the end, because the shorter bands move faster. And then so there's a lot of applications for that, as you can imagine. So you can determine sort of length of DNA and stuff like that. Hopefully that makes sense. Okay, cytopol. Again, I know this is a lot of information being thrown at you. Just try to digest it as best as you can. Okay, ranking. So the correct order of the steps for PCR. So do you guys remember what the degrees that you need are? Okay, won't give you too long. But yeah, okay, looking good. So yeah, denaturation, annealing, elongation. So really hot. And remember, you have to know the purposes of these. It's easier for stuff like this, like and things with bio, and again, like literally all your subjects, try your hardest to not just rote memorize stuff. So think like, okay, 95, 55, 72, 95, 55, 72, and just try to remember it like that. Have a deeper understanding. Why do I have to warm it to 95? I have to get it really nice and hot so that you can break the hydrogen bonds and separate the double strands. Why do I have to lower it? So that their hydrogen bonds can reform when we're thinking of our primers attaching. Why do I have to raise it again? Think of, yes, Taq polymerase. It works really well under heat. This is a good temperature for it to work nice and fast. Like think about that with the processes of PCR, and that's a lot better, and that'll aid your understanding and give you a much deeper understanding than just rote memorizing, okay, 95, 55, 72, and trying to remember it like that. Okay. I think, let's see if there's any more. Okay, so there's a little bit more left on DNA manipulation. I'll leave this. So it's the use of recombinant plasmids to transform bacterial cells. This is a bit of a complex process as well. Again, you'll get it. I know I'm saying this a lot, but like have faith, you'll get it. After you look at it a lot of times, you'll get it. So in terms of plasmids, basically in a bacteria, we have a lot of chromosomes. They have one circular chromosome. So just one circular chromosome. They also have these other little tiny sections of DNA called plasmids, and then just like little small extra circles of DNA. So they're a circle. They're not strands, long strands like ours. They're a circle. An important characteristic of this is that bacteria can share plasmids. So they can take up plasmids and I guess like send them out as well. So we basically use these plasmids, again, as vectors to insert genes that we want to be inserted into bacteria, and then bacteria will produce these. So for example, with insulin, we insert the insulin gene into a plasmid using our little tools. We then put that plasmid into a bacteria. Remember, a really big thing about bacteria is that they replicate super quickly. So you put our little basically human insulin gene into this bacteria. This bacteria will divide and divide and divide and divide really quickly. And then you have a bunch of insulin producing bacteria. They're producing the insulin. You then harvest that insulin, purify it, and then you have human insulin. So that's the general process. This will probably help you. That's what it looks like. So I've explained the general process in terms of how this happens. So you have your plasmid. This is just a plasmid. It's just normal. You cut it with our endonuclease. Remember our little scissors? They just chop it up. We get our little open plasmid now. We then get the gene of interest. So this is our human insulin gene. You have to cut it with the same restriction enzyme. Think, why would I need to cut it with the same restriction enzyme? Why can't I cut it like it's just scissors? Remember, these scissors have very specific recognition sequences. You can almost think of it as like, it's not, like the scissors that you used in primary school and they had like zigzags or they had like waves, like little patterns like that. Think of that. You can't attach paper that you've cut with a little zigzag scissor to one that you've cut with a wave scissor. It doesn't match up. You have to cut zigzag scissor with zigzag scissor so they can fit in nicely. Think of that. So you cut your human insulin gene with the same endonuclease. So then think of sticky ends, they'll attach really nicely. So basically you put them together. So you get your human insulin gene, you whack it in that plasmid, and now we've got our recombinant plasmid. So this is an important part to remember. Recombinant plasmid, because we've like recombined the genetic material basically. So something else that we do is we add antibiotic resistance into this gene as well. So we've got this nice plasmid, we've got insulin as part of it, and we've also got a gene for antibiotic resistance. So we've got that in our plasmid now. So we will then use our recombinant plasmid, and we need to get this into our bacteria if we're going to produce our insulin, right? So don't really worry about this too much, but basically you just force the bacteria to take up this plasmid, and then you will use that antibiotic resistant gene to determine which bacteria have taken it up and which haven't, because some bacteria just won't take up the plasmid. So that's why the antibiotic resistant gene is really important as well. And also sometimes, the plasmids may not take up the gene, stuff like that. So basically that's why the antibiotic resistance is needed. It's like a little tag to say, which bacteria have got the plasmid that contains the insulin gene in it. So we have the bacteria, we put our antibiotic, obviously ones that don't have the antibiotic resistance gene and therefore don't have the plasmid, they'll die. So goodbye, whatever's left obviously has got that antibiotic resistant gene, and that's got our insulin, and therefore we will then grow those and we'll get our insulin from there. Hopefully that kind of makes sense. Okay, so now moving on to GMOs. So basically GMOs and TGOs, genetically modified organism versus a transgenic organism. Think about the names. So genetically modified means anything that has had its genome altered in any way. So they've had a gene removed, they've had maybe a gene like swapped around or something like that, or you've kind of added a gene from maybe like a different organism of the same type, like the same species. Think about it, something like that. Genetically altered in any way. In a transgenic organism, this is a specific type of GMO. So understand that transgenic organisms are also genetically modified organisms. Think of it like an umbrella, because a transgenic organism, think of transgenic, you've got a gene that's come from another species. So that's being put into another organism. And then so obviously, you're going to show that gene, whatever the gene program's for, you're going to express that protein, express that characteristic. So therefore it's genetically altered. So it's a genetically modified organism. Something important to understand, which is in this last dot point, all transgenic organisms are genetically modified, right? Because their genes have been modified. However, genetically modified, if you are a GMO, a genetically modified organism, you're not necessarily a TGO. So if you have a plant and this plant has a gene that makes them susceptible to a disease, you take that gene out. It's genetically altered. It's a genetically modified organism. It is not a transgenic organism because it hasn't had a gene from another species inserted into it. Hopefully that makes sense. That is such a common thing that can strip students up. Just think of it like an umbrella. GMOs are the umbrella. TGOs are just a specific type of GMO that can fit into that category. Okay, in terms of cotton, so cotton is basically just an example of a TGO, I think. Yeah, so TGO, again, it's just like a certain characteristic. So this one here, a natural insecticide against caterpillars. So basically makes them not susceptible to these certain caterpillars. You can have other TGOs that might be for a certain aesthetic characteristic, or again, like growing bigger fruit or crop yield, stuff like that. It's important to think about ethical conditions as well. With the new study design, I would say there's an emphasis on ethics, especially with research methods. So with your area of study 3 stuff, bioethics is being pushed a lot more. It's not so specifically outlined with GMOs and TGOs as it has been in the past, but because there is such a big emphasis for for, you know, kind of ethics in general with area of study 3, it's important to kind of think about this stuff as well. Okay, so you can think about things like this. So ethical stuff, obviously, social considerations, biological considerations. So think about if you've got TGOs, if those were to, you know, reproduce with other non-genetically modified things, then you've kind of mixed stuff up. Thinking about artificial selection, think about, you know, future generations of that species, stuff like that, how they might be affected. Sometimes it's this idea of should humans be meddling around? Socially, think about economics. So if you've got a TGO and it's producing, you know, really big fruit and whatever stuff like that, people are all going to want that. So it's kind of leaving other people in the dust. Other things like that as well. Take them into consideration. This